Okay, good morning. First, I want to go over the worksheet that people have been complaining about, and then just a reminder about general cloning and how cDNAs are going to be helpful to us. So it should be a pretty easy day overall. Oh, okay. So I'm showing you the first 60 nucleotides of the hemoglobin cDNA, the human one. And as usual, there's only one strand shown, and we have to imagine the other strand. Now, the strand shown is running 5' to 3'. And what we're trying to do now is design PCR primers that will allow us to amplify this short piece of hemoglobin cDNA. Okay, so the first thing we have to remember, and that's really crucial, is we're going to have a forward primer and a reverse primer, one for each strand. And it's important to recognize the orientation because that tells you what sequences you need. In the picture we have the two primers need to have their 3' hydroxyls pointing across the region we're trying to synthesize so that DNA polymerase can then extend those primers and create the new strands. So in my picture, I've simply drawn primers that will then satisfy those criteria, and that includes one that's 5' to 3', that's our forward strand, and the other is 5' and 3' in the reverse, and that's our reverse primer. So if you look at the configuration of these sequences, you realize that your forward primer is going to have to equal in sequence the upper strand, because that's the one that directs the 3' hydroxyl in the correct direction, and that the reverse primer is going to be the complement of the strand that's given, because that's the one that has its 3' hydroxyl correctly oriented. And notice that that reverse primer then matches the lower strand in sequence, equals it. Okay, so with this, I've just written in the sequences, and you can see that the forward primer I've written down next to the, base paired to the non-visible lower strand. And you can see that that primer complements the lower strand, and that it equals in sequence the upper strand. And the same is true of the reverse primer, where we have the 5' to 3' with its 3' hydroxyl pointing towards the left. This will bind to the upper strand and is equal in sequence to the lower strand. So that's the key thing you have to remember when designing primers for anything of this sort. Okay, so in our hemoglobin example from our worksheet, we just used a 10 nucleotide primer, which is way too short in reality, but it makes it a little easier to look at. That when we're looking at a sequencing reaction, we have to figure out what primer is going to bind where to go across the template as above. But in the sequencing question, we're going to then ask what that sequence is then going to look like. So that's where it gets a little bit tricky to think about, because this particular primer equals the upper strand. It will bind the lower strand as a template, and will continue to use the lower strand as template. So that's the key idea with sequencing. That lower strand is going to be the template for production of essentially a new upper strand. So I've underlined the first 10 nucleotides of the newly synthesized DNA, and what that will be colored according to whatever your nucleotides are in the dideoxy reaction. But importantly, that is the sequence that you will generate by copying the lower strand template, and it will have the colors because the template is going to be copied with the possibility of having specific stops at each of those positions in the usual sequencing kind of reaction. Okay, so here's our picture again. We need to look at the reverse primer now in the reverse reaction. And it's the same idea, but you need to think about it a little bit differently because of what you're actually given in the slide. So in this case, our primer equals the complement, rather, of the strand that we're given. So our primer is not the one that's written. We have to make the complement of that, and then it will have its correct 5' to 3' orientation pointing into the region that we're trying to synthesize. Right. So again, we're going to be having something that's using that strand. In this case, the upper strand is going to be our template because that's the thing to which it's bound. And then when we go again synthesizing across that insert, the first 10 nucleotides are going to be the complement of that template. And that's what I've got underlined here. And that's the answer to what the first 10 nucleotides of the sequencing product are going to look like. So getting the forward and reverse primers right is the first step. But then you have to envision the synthesis reaction that's actually going to occur in response to those primers to predict what the um, colors of the trace of the peak is going to be. So it's a little bit, there's two layers to it, and the second step I think is a little bit more difficult. But just draw it out for yourself and convince yourself. Okay. A few minor issues I had with this. Polarity is in a nucleotide direction. It's not the negative charge. A lot of people wanted to say that yes, the polarity is negative, which in a chemical sense, of course, that's true. It is negatively charged. But when we're talking about nucleic acids, what we mean is 5' to 3' or 3' to 5'. It's just a different use of the same word, and it refers to the direction of the sugar phosphate backbone as you go along the molecule. Okay. And the last part, people did rightfully describe the fact that the two polymerases might collide if you had both sequencing reactions going on at once. But the answer I was really looking for is that if you had two reactions going in opposite directions with different templates, essentially, you would have two colors at each position, which is to say two peaks at each position, and that would give you an unreadable sequence. Okay, so for further reinforcement, there's a new worksheet which I posted, and I will go over this next time, and it will be due on Friday. This is just a PCR type question without sequencing thrown in, so it'll be a little easier to understand. But what we're going to be doing is analyzing the results for different alleles of this particular STR locus. So we'll do this one in class next time. Okay, we're going back to our basic cloning reaction with simple plasmids and then talk about some cDNAs. So back at the very beginning, back in Cell-and-Moleq, we envision what your basic cloning reaction is using a plasmid cloning vector. So we cut the plasmid cloning vector with a restriction enzyme, which cuts it at one specific location in the molecule. And then at the same time, we've prepared a double-stranded DNA to be cloned, and it is a single piece of DNA which has been cut with the same restriction enzyme. And then in vitro, we combine these two with the enzyme DNA ligase, which is able to restore the phosphodiester backbone in those two molecules, attaching them to each other, so that now when we created this now recombinant DNA plasmid, which is partly bacterial origin and partly something else, let's say human, is now an intact molecule that can be transformed into E. coli for further propagation. Okay, so things the vector has to have: an origin of replication to propagate it, a selectable marker for transformation, and at least one unique restriction site that we can use to open the molecule for the addition of the insert. So more typically in plasmid cloning vectors, you have a multiple cloning site region, which has sites for numerous enzymes so that one has a choice as to which enzyme one's going to use. So here's the simplest possible cloning vector, and it has essential ingredients: drug resistance, origin, and then a schematized multiple cloning site, which has sequence sites that enable unique cutting for any one of those particular enzymes. Okay, here's a detailed view of one that's in the PUC118 cloning vector, and it has sites for all these enzymes that we can see, and the sequences are given. So the insert, of course, is a piece of DNA that's going to be carried. It might be a piece of chromosomal DNA, or it might be cDNA copies of mRNA molecules, as we'll see in a bit. So in terms of how this process goes, we certainly need to know something about some of the enzymes, DNA ligase for sure, but also some features of some of the restriction enzymes that might be want to use. So restriction enzymes, remember, these are sequence-specific endonucleases. So the endonuclease refers to the fact that the enzymes cut DNA internal rather than chopping off at the ends. So it's going to be inside a double-stranded molecule that it's going to cut. And the fact that they're sequence-specific means that they only cut the DNA at or near the location of their specific recognition sequences. So what they do is they cut both strands of the phosphodiester backbone at or near their recognition sequences and nowhere else. So the basic cloning reaction has us cut the vector with a restriction enzyme, cut the insert with the same enzyme or one that generates the same sticky ends, mix them together in a test tube so they are allowed to base pair. Then, in their floating around, they will encounter each other, transiently base pair, and DNA ligase will then seal the nicks, thereby attaching the two molecules together. After that, we'll transform them into E. coli cells, selecting for drug resistance. Okay, so some important points about restriction enzymes, besides that they're sequence specific. They differ in what their recognition sequences actually are, the length, but also the actual sequence. And so they will produce different types of sticky ends according to what the actual recognition and cut site might be for that particular enzyme. Okay, so the example everybody's familiar with, this is one, EcoR1. Its sequence recognition cut sequence is GAA-TTC. Now, usually they don't give you both strands. You have to write in the second one yourself. And this is running 5' to 3'. And I put the little asterisk on here to indicate the location of the cut site where the phosphodiester backbone is cut by the enzyme in response to this particular sequence. Okay, so what happens when you cut the molecule here is that the two halves of the molecule will fall apart. They're still transiently base paired initially, but those 4 nucleotide base pairs is not enough to hold the molecule intactly together at room temperature. So the molecule will flop open, revealing the sticky ends, which are at the 5' ends of the two strands. So the 5' end reads 5' AATC, and then the GC and CG are base paired, etc. So this molecule can flop back together, or it can flop in solution and acquire another AATT series of base pairs from an insert, or another copy of itself for that matter. So these are called sticky ends because they're available for base pairing, and they are at the 5' ends of the molecule. So these unpaired regions of the cut molecules are going to be what's going to associate with the DNA to be cloned. So while we're at it, other restriction enzymes have different cut sites. BAM is GGATCC, Hindi3 is AAGCTT, etc. These sequences occur more or less at random in normal DNAs. Okay, here's the double-stranded structure for BAM, etc. Here's some examples from an earlier textbook which shows you different enzymes. This illustrates the fact that different cut sites can be different in length, in fact, with some cutting at 4 base pair sequences, some at 6, and some at more than 6. And it shows you the overhangs that result from each of those digestions. Okay, so the idea of a cloning reaction is instead of having the molecule flop back and reattach to itself, we're going to try to get it to attach to something else. And once DNA ligase has gotten there, it will enable it to restore or create an intact phosphodiester backbone, which will keep the molecule together. So here's a very, very basic sort of picture. The arrows are the indication of where the cut sites are for EcoR1 site. So the arrows indicate where the molecule will be cut by the enzyme, allowing the two halves to sort of flop away from each other. So this is what the sticky ends will look like after they flopped away from each other. These single-stranded bits are now available for base pairing, either to each other or to something else coming out from the solution. So meanwhile, we've prepared our insert in such a way that it has sticky ends which are compatible with or identical to the cut well, the vector that's been cut with EcoR1. So they're going to be compatible, meaning that they can base pair and then be ligated. So then they're going to find each other in solution and then be ligated by the ligase. So this is a picture of a piece of DNA in black with sticky ends that match the cut EcoR1 site. So in solution, that's able to associate with the cut vector molecule, base pairing, transiently. It could then flop apart. But that's what the ligase does for us. It reattaches the two, where it attaches, creates a new phosphodiester bond at the locations where the nicks occur in this particular template. So we're only showing the desired reaction. You get other things that happen as side products. Inserts can ligate to other inserts, vectors to vectors, etc. but many of these we're not going to be able to transform cells. So we're going to skip the niceties of that right now. Okay, so in transformation, the DNA is taken up from the outside, and in the way we do it in E. coli, the phenotype will change from ampicillin sensitive to ampicillin resistant. So these cells that are able to take up the exogenous DNA that are called competent cells will be treated with the plasmid and allowed the opportunity to take up the plasmid. Now, this is not an efficient process by any means in E. coli, and that's why we need to have the antibiotic resistance gene to select those cells which have acquired the plasmid. In some species of bacteria, this transformation process is quite efficient, but in E. coli it's not. And so we have to have this selection to make sure that anybody that lives and survives the drug will contain a plasmid molecule and be transformed. In standard DNA cloning, restriction enzymes are used to produce sticky ends, synthesize the insert, add nucleotides to the end of the insert, remove restriction sites, create covalent bonds between the insert and the vector. Right, A, we produce the sticky ends. The role of DNA ligase in molecular cloning is to synthesize the insert, attach the vector to the insert, align the complementary sequences, sticky ends, digest the DNA at the restriction site, replicate the molecule in E. coli. Yes, B, attach the vector to the insert. Okay, so now the real business of the day, we have to decide what to clone. In the example being cDNAs, why bother doing that when we can just get a chunk of the mammalian gene? Well, it turns out it doesn't work that way, not at all. Mammalian genes are for sure very large, and they also have a lot of large introns. And the pictures they show us in textbooks are usually not very informative of how large those introns can really be. And E. coli, of course, does not have this type of spliceosomal introns and cannot remove them. So even if we could get the whole gene into a plasmid and we got it into E. coli, we couldn't produce a protein from it because E. coli doesn't know how to splice introns. Okay, and the other thing is that the gene is usually too large to be carried by a plasmid. Okay, so here's two human genes just to show you how extremely different they can be. The first one's a human beta-globin. It's really quite small and only has 3 exons. And the other is, granted, another extreme, the human factor VIII gene, which has 26 exons and spans more than 200,000 nucleotides. So that 200,000 nucleotides, that's 200 kilobases, that is way too big for an E. coli plasmid to carry under any normal circumstances. Also notice that the introns are considerably larger than the exons. And this picture isn't even really to scale. The little red stripes are the exons, and the really large, long orange things are the introns. So most of this gene, long gene that it is, most of the primary transcript made from this gene is going to be spliced out in the process of putting those exons together. Remember, the exon is the part that's kept after splicing. Okay, so this is, of course, extreme, but in fact, the average human gene is still a lot bigger than the exons. Okay, so the average gene, on average, has about 9 exons, and the final length of the total mRNA is about 2,500 nucleotides on average. So on average, actually, the primary transcripts are much longer than this, and that more than 90%, or approximately 90% in some cases, are actually spliced out during splicing. So most of the primary transcript is actually discarded many times. And even within the 2,500 nucleotides of the mature mRNA, some of that is not actually coding region. We have 5' UTR, which is before the AUG, and we have the 3' UTR, which is after. So the average protein coding region of the average mature mRNA is only about 1,400 nucleotides. Now, those mRNA links fit nicely into a plasmid vector for the purpose of producing a protein, whereas the entire gene itself really is too big, and we can't get rid of the introns anyway. Okay, so taken together, producing the primary transcript in prokaryotic cells would not help us because we'll have the introns there. We don't have spliceosomes to remove them. And so we need to take another path in order to produce the protein in E. coli cells. So this is a reminder of what happens in a eukaryotic cell, the path between the double-stranded DNA and the actual protein translation in the cytosol. So of course we have transcription beginning at the promoter that goes across the double-stranded DNA sequence. That's transcription. Then we have various modification events or processing events where the primary transcript, which is the one that's collinear with the DNA, has its introns removed. It's also polyadenylated and capped. And then finally it's ready for export to the cytosol where translation will happen. So when we're talking about cDNAs and cloning them, what we're talking about is this final product, that's the mature mRNA in the cytosol that has its protein coding regions lined up nicely so that translation can occur. So this is what our cDNA will be used. Well, this will be used as a template to produce a cDNA because that's a more convenient form of cloning and capable, at least potentially, of producing the protein directly in E. coli or in a mammalian cell. This says the same thing basically. We've got our gene. Our introns are, of course, ridiculously small compared to real life. After transcription, the RNA is spliced, and that RNA is then sent to the cytosol for translation. So that's the business part of the protein of the mRNA. Okay, this is another picture in slightly more detail with a few more things that we don't need to worry about quite yet. We have discussed the promoter already as something preceding the gene, which is not going to work in E. coli anyway. And what we're looking at right now is simply this mature mRNA, which is capped and polyadenylated and has been spliced so that the codons are there in the correct register so that presenting them to the ribosome would enable the production of the desired protein. So I just summarized all this. Promoters bound by RNA, pol, etc. So what is the cDNA? Cloning cDNAs instead of mammalian genomic DNA helps a lot. Okay, these are DNA copies of mature mRNAs. They're made enzymatically in a test tube and then cloned. Okay, so what it includes is the DNA version of the mRNA sequence, no introns, but it has no promoter either. So whatever's left over after splicing is what's going to be there in the cDNA. Okay, so creating a DNA version of an RNA is not a trivial process. It involves numerous enzymes. Importantly, to realize that what you want to do is take an RNA, use it as a template to produce a DNA. Well, the DNA polymerase, the standard normal type, cannot do that. And so the enzyme that's critically important to this is reverse transcriptase. Okay, so this is a RNA-dependent DNA polymerase, and it's made from retroviruses. And this is a normal part of the retroviral life cycle that we've exploited for our own purpose in making DNA copies rather of mRNA. So they're like DNA polymerases in that they require primers, but we can give them primers. They can use oligonucleotide primers, just the way we would use primers for PCR or sequencing. Okay, in reality, they will also use RNA primers, which is what they do as part of their own normal life cycle. So we're grabbing this enzyme from retroviruses and using it to create DNA copies of cellular RNAs that we're interested in. Okay, so don't get too bogged down in the details here, but it's important to understand the enzymology a little bit. Okay, so our first picture shows our mRNA, and it has a poly A tail. Well, we know that we can use an oligo DT primer, which is to say a primer which is simply a string of T's, which will bind to the poly A tail at the 3' end of the mRNA, and then reverse transcriptase will then use that primer and extend all the way across, if you're lucky, the mRNA sequence. So that's your first strand cDNA synthesis, where your DNA primer essentially has been used to prime synthesis of one strand of the DNA in response to the mRNA template. Okay, so now we've got this hybrid, really, mRNA cDNA. What we need to do is get rid of the RNA, and for this purpose, we can use RNase H to make some nicks along the template. And then the DNA polymerases, because now you've got a DNA template. The first strand you made is DNA, and these little bits of RNA can serve as primers for the DNA polymerases that have the 5' to 3' exo activity. Then the DNA ligase can be used to attach those little bits of DNA together. The ends are blunted, and now this double-stranded cDNA is ready to be cloned. So what you need overall, in summary, you need your RNA template, you need reverse transcriptase, you need a primer. In this case, we're going to use an oligo-DT, which is an oligo-DT 18 long, a little bit of RNase to digest little bits of the original RNA, and DNA polymerases, whatever. So now you've got this blunt end molecule. Now what are you going to do with it? You've got to clone it into an EcoR1 cut vector. Well, you got a problem here because it has no EcoR1 site on it. Okay, so one easy workaround for this sort of situation is to use PCR to add sequences to the ends that will allow it to be cloned into whatever vector you want, using whatever sites you want. So, okay, we arbitrarily chose EcoR1 for this, but if your sequence had EcoR1 sites, you probably wouldn't want to use that. So we want to pick something that does not cut within the cDNA sequence. This is just for the sake of argument. So adding restriction sites to your primers means you're going to have a section of the primer which acts like a normal primer. It's going to be complementary to your template, and then it will be able to prime synthesis across to generate a new strand. And the same is true of a reverse primer. You've got something that will anneal and allow extension across the gene of interest or the cDNA of interest and create new strands. Okay, what's special about this is that in addition, we will include restriction sites in the middle of each of those primers. They can be the same or they can be different. Okay, and you need a few extra nucleotides on the ends because restriction enzymes don't like to cut on the extreme ends of molecules. So this extra bit, you don't want, you probably should, but it's a little complicated to follow the series of reactions that if you use these primers, generate something which goes all the way from one end of the forward primer to all the way at the end of the reverse primer to give you something which is double-stranded completely, but with restriction sites on the end as determined by the primers. So we've got a few extra nucleotides, etc. So what this looks like in this particular scenario, you've got the added stuff. Now this doesn't make it difficult for the primer to anneal, the real primer, because you've already got enough here to anneal the primer. But what happens as a course of the reaction, when that original strand gets copied, that primer sequence is there. So you end up generating long molecules which contain the added sequences that you put on with the restriction sites, as well as what was there in the first place. So don't worry about too much of the details here. Just take my word for it. This does work, but it's worthwhile to work through the strands to see exactly when that happens and how it happens. But this is eminently clonable now. You've got a restriction site in the exact right place that you want for cloning purposes, and that's the point. Right, so here's what it looks like. Your PCR product will have an EcoR1 site, insert, and another EcoR1 site with a bunch of sort of random nucleotides at the end. So when that's cut with EcoR1, the middle piece with your insert falls out, and so that's ready to be cloned into a plasmid vector or whatever. So there are gazillions of things available commercially, gazillions, and they're quite happy to sell you or even clone something for you if you want. Not literally gazillions, but several 100,000 cDNA clones from many, many organisms are available for purchase. So most people don't actually make their own cDNAs, but they might get the cDNA and then flip it into a different vector or something. Okay.